Hello and welcome back. In this video we're going to finish the cell structure and function. This will be part 2 video, and in this one we're going to talk about the cell membrane and how it's structured and what it does in terms of its function. The cell membrane is a really important part to the cell, and that's why we spend a lot more time on this particular part. And throughout the course of anatomy and physiology we'll talk about the cell membrane and for a lot of different lectures and a lot of different systems. So the cell membrane is a universal component of all cells, so it's found in every cell of your body and in every cell of every living organism. And it has certain parts to it that you'll be responsible for understanding. So let me talk about those for just a second with you. So the cell membrane, you can see over here is a cell membrane. It's a bilipid layer. It's made of the phospholipids, which remember has a hydrophilic head and two hydrophobic fatty acid tails. So the head structures affiliate with water. The tailed structures, the fatty acids, are basically shielded from water in the interior. And it's made of two sides. You can see this is one side of the membrane. This is another side. That's why it's called a bilipid layer. So the phospholipids, remember, contain hydrophilic and hydrophobic regions, and that gives it the ability to form this particular layer. The hydrophilic heads are made of glycerol, and the hydrophobic tails are made of fatty acids. Now those fatty acids can be saturated or unsaturated. Saturated fatty acids, remember, don't have any double bonds, and this makes a very viscous, that is a slow-moving type of cell membrane. Unsaturated fatty acids, though, do have double bonds. They have kinks in them, and they make the cell membrane a lot more fluid. So the cell membrane is a fluid. A lot of people can't understand how can you have a fluid that makes a membrane, but it is a fluid. It moves very slowly. It has the properties of a fluid, but it does have parts that anchor it to the underlying structures, so that it doesn't just completely flow off. It also has other parts inside of it that help it to maintain its structure. So another part that's associated with the cell membrane are the proteins. We have integral proteins. These integral proteins are going to go through the whole membrane. So you can see this is the protein here. Here's the phospholipids on either side there. But you can see the membrane expands through the whole. This protein expands through the whole membrane. So it does have a hydrophilic region, so it has amino acids making up this protein that like water, and it has a region that will associate with the water that's on the outside and on the inside, and it has hydrophobic regions where you have nonpolar amino acids that don't affiliate with water in the interior. There are also peripheral proteins. Peripheral proteins are going to be on the outside surface. Peri means around. So on the outside surface you can have some of these proteins, and here's one right here. What this protein's doing in this particular example is anchoring the cell membrane to the cytoskeleton. So since you have anchorage of the cell membrane, you can do things like this. You can change the shape of the cell membrane. You can do phagocytosis with the cell membrane, all because you have these anchor points that are made from proteins. So these proteins that are peripheral can be anchored to the cytoskeleton, or they can also be anchored to the integral proteins as well, for causing different various things to happen. Well, these membrane proteins serve lots of different functions. They actually help to transport substances into and out of the cell, such as sugars or ions. They have enzymatic activity where they can help to break products down. You have a lot of this in your intestines, enzymatic activity. All the food you eat, is broken down to its simplest building block by integral proteins that are serving as enzymes in your small intestine. They do have the ability to pick up signals. So here's a signal molecule coming down, and these proteins have the ability to transmit the signal to the interior of the cell. They're also responsible for helping cells join together so you can form sheets of cells and therefore tissues. They do recognize, so these little proteins and other structures on the surface help in cell-to-cell recognition. Okay, so when you get a blood transfusion of the wrong person's blood, your body will recognize that that's foreign and fight it. But if you get the right blood type, then you can actually recognize that and not fight against it. There's also, these proteins also serve for the attachment of the cytoskeleton to the membrane and the extracellular matrix that we'll talk about in just a second. Cholesterol is another important of your cell membrane. It's pretty important. You can see how it's located down here in this little graphic here. So there's your cholesterol serving the role. And what cholesterol does is it helps at high temperatures, it helps the membrane to decrease its fluidity, so that at high temperatures the membrane doesn't become too fluidy and flow off and the cells just open up and release all of its contents. At low temperatures, it will not allow the membrane to become a solid. So cholesterol is almost like a buffering molecule. It buffers high temperatures and low temperatures to keep the cell membranes, performing at the doing the job it's supposed to do. You oftentimes see cholesterol more associated with animal cells. So when you eat any kind of animal product, such as an organ or muscle tissue, you will be eating cholesterol with that. Plants, not so much. So carbohydrates are also associated with the cell membrane. These are proteins or lipids that have carbohydrates attached to them. And some of our really complex proteins on the surface include proteoglycans. We'll talk about some of these molecules later on. Glycoproteins, these are sugar proteins. And glycolipids are, sugar lipids. They have sugars attached to lipids. Collectively, these molecules make up what we call the glycocalyx, and so it surrounds the whole cell, the whole outside cell, and coats it. I think of the glycocalyx as almost like the cell glue. It helps cells to join to each other, but it does other very important functions other than that. So it lubricates and protects the cell. It anchors and causes the cell to have the ability to change its shape and to move. The glycocalyx also serves that job of recognizing self versus non-self, and it has receptors in the surface of it that can receive information as well. And collectively, this is what the glycocalyx looks like. You can see here a glycoprotein, and so here is a glycolipid, so these are other kinds of proteins we'll talk about when we get into talking about tissues. Okay, but it's kind of like a glue, or it serves other functions, lubrication, reception, and those kinds of things for the cell. Now, cells can be joined together in several different kinds of ways. I think you'll have to know 4 different kinds of ways they join together. One of the ways is called a tight junction. This is a junction between cells where the membranes between adjacent cells actually fuse together. So you make an impenetrable barrier between those cells that are adjacent to one another. Tight junctions, a place you see a lot of tight junctions would be like the urinary bladder and the intestines, because you don't want urine leaking into your body, and you don't want bacteria coming into your body through gaps in cells. Another really important type of intercellular junction is the desmosome. Desmosomes are like cell rivets, and they basically fasten cells together in strong sheets. So if you ever take and pull your skin, you can't pull your skin but so far. I'm just trying to show you that you can't pull your skin but so far, because these desmosomes hold those tissues together very tightly. Another a little bit more rare junction is called a gap junction. These form cytoplasmic channels between cells, so they're like little holes that are between cells, and they're very common in animal embryos, because there has to be a lot of communication between cells so that you can form the right, appendages and the right organs. But you also see these in adult cells in heart muscle cells. Heart muscle cells have the ability to conduct electrical signaling through those gap junction channels, so the heart will beat in rhythm. Hemidesmosomes will anchor cells, so they're anchor points to underlying cells or underlying things like a basement membrane, a proteinaceous layer that many of your types of cells sit on. So these are the 4 types. You have your tight junction, that fusing of the membrane so things can't leak between cells. You have your desmosomes, which are the things that rivet cells together. You have your your gap junctions, which allow for signaling to occur in between cells. And then you have your hemidesmosomes that anchor cells to underlying things. In this particular example, it's anchoring it to this little extracellular matrix of another cell, but it could be a basement membrane, a non-living proteinaceous layer that many cells sit on. Now, the model we use for the cell membrane is the fluid mosaic model, and that basically describes that the membrane is like a fluid. It's like salad oil, made up of a phospholipid bilayer, but it's also a mosaic of parts. It's made of proteins and phospholipids and cholesterol. So it's a mosaic of parts, but it also is a fluid. So the fluid mosaic model looks like this. We used to think of it as being like a peanut butter sandwich with layers of bread, but we now know that the proteins can move, they can spread apart, and it's a mosaic of parts. But it does flow like salad oil. Now, a good question to ask is, how do things come into and out of the cell membrane? So the cell membrane happens to be selectively permeable. So it's permeable to some things, but selectively not to other things. So hydrophobic substances can go right on through the cell membrane. Hydrophilic substances cannot. So proteins are used to bring in hydrophilic substances. Now we have different kinds of transport depending upon if you use energy or don't use energy. So passive transport is a type of transport that does not require ATP or cellular energy. One type of passive transport is diffusion. Diffusion is where you have a population of molecules that move from an area of low concentration to an area of high concentration. So, an area of high concentration to an area of low concentration. So higher concentration to lower concentration. So I'm just putting some little dots here on the screen for you. So imagine if someone sprayed perfume in the room. The perfume is very highly concentrated here, but eventually it would move all throughout the room and eventually be in equilibrium, all throughout the room. But that's diffusion. Diffusion occurs because molecules run into one another and then they begin to spread or work their way out from where they're running into each other. So diffusion, it's passive, and it moves from an area of high concentration to an area of low concentration. So there are substances that we bring into our cell that uses diffusion exclusively. For example, if you have these dye molecules here, you can see here's a selectively permeable membrane. Over time, you can see that if they can go through the cell membrane, you can see that eventually there will be an equilibrium. So they're diffusion from an area of high concentration to an area of low concentration. So here's showing you that it can go both ways. So if you have a high concentration of the orange over here, they can work through and eventually spread themselves out. If you have a high concentration of purple, it can work in the opposite direction and move themselves over to the other side. Some things that use exclusively diffusion would be like carbon dioxide, oxygen, and various kinds of hydrocarbons. These are things that don't have a charge, they're nonpolar. So make sure you know that. That's oftentimes a question people miss on a test, or what are some simple substances that can diffuse through the membrane, and that would be carbon dioxide and water and hydrocarbons. So there are things you can put on the surface of your skin that are medicines that will actually go through your skin because they can penetrate through cell membranes. Other kinds of medicines you actually have to ingest. Osmosis is a facilitated diffusion, which means it's the diffusion of water, but it has to actually go through a protein channel. These protein channels are called aquaporins. And water moves from an area of low solute to an area of high solute. Okay, so if we had a little beaker right here in a semi-permeable membrane, let's say over here that this is salt water over here, and this is pure water over here. So this would be salt water over here, and this would be pure water over here. Okay, so by definition, water moves from an area of low solute, which is this side over here is low solute. It moves from low solute to high solute. Okay, so it moves in this particular fashion here. So that's the diffusion of water, moving from low solute to high solute. And you probably have seen this before if you've ever put salt in a slug before. Salt is like pure solute. So if you put salt on a slug, the salt will draw the water out of the slug, thus killing the slug. Okay, because the water's moving from an area of low solute from inside the slug to an area of high solute or into the salt. If you drink salt water, the same thing happens to you. The salt will draw water out of your body, and then you'll have liquidy diarrhea, and you'll die. So we have to be very careful about what we ingest. And this is just showing you different kinds of solutions I'll talk about in a second. But here's a solution over here that we'll call it A. A is a solution with low solute, and B is a solution with high solute. And you can see the semi-permeable membrane in between. Water will literally move that way, okay, and fill up the other side of that bent tube. So there are different kinds of solutions when you compare them to each other. So when comparing two aqueous solutions, three states can be observed. You can have a hypotonic solution. A hypotonic solution is a solution with low solute concentration compared to another solution. It'll have greater amount of water because it has lower amounts of solute. A hypertonic solution is a solution where you have high solute but less amounts of water compared to another solution. And then if you have two solutions that have equal solute and equal water concentrations, we call that an isotonic solution. Okay, knowing these solutions are really important in anatomy and physiology, and if you're going into the medical field, this is something you really would want to definitely know about. So here we have an isotonic solution, and what I'm going to show you is what happens to a red blood cell in different kinds of solutions. So here's your red blood cell, and here's an isotonic solution. If you notice the arrows, the molecules are moving in and water's moving in at the same rate that it's actually moving out. Okay, so you have a cell that maintains the right cell shape. It's a healthy cell, and there's nothing wrong with it. In a hypotonic solution, though, you got some issues that are going on. Because you have, in a hypotonic solution on the outside, you have more water that's running into the cell, which has more solute on the inside. So in a hypotonic solution, water rushes in, and when water rushes in, it's going to cause the cell to swell up, as you can see in the scanning electron microscope picture down here at the bottom. Okay, those swollen cells can burst, so you definitely wouldn't want that to occur, because then everything that's inside the contents of those red blood cells leaks out, and we know that the plasma of the blood has certain levels of ions in it, and if you release the ions inside of blood cells, that can upset the balance of the concentration of ions. That causes damage and destruction. In a hypertonic solution where you have a lot of solute on the outside, you can see the water moves out of the cell, and you get cells that crenate or shrivel up, and that can be bad as well. So just a question for you after I tell you this definition. Osmoregulation is the control of water balance by organisms. So every cell in your body osmoregulates. It has very specific mechanisms to make sure or ensure the right amount of water is on the inside of it. So what type of solutions do you think are IV fluids? Are IV fluids hypotonic, isotonic, or hypertonic? Why do you think that? Well, what we've discovered when we studied animals and started working out creating these various solutions is we figured out that IV fluids have to be a 0.9% sodium chloride. Okay, they have to have some dissolved solutes inside of it, and so that it can mimic what's inside the plasma of your blood. So your plasma of your blood is about a 0.9% solution, and so to be isotonic, that is, so water can flow into and out of your blood cells at the same rate, IVs have to be slightly salty. So an IV can also sometimes add dextrin, which can help to make the solution isotonic. Dextrin is a sugar, so you can actually eat that sugar as well. So it not only does it the IV rehydrate people, but it also can feed people sugar at least. So facilitated diffusion, this is the diffusion of materials using proteins, but not the diffusion of water, but the diffusion of ions and other molecules using proteins. So we do have channel proteins. Channel proteins are little proteins that sit on the surface of the inside the membrane, and they can, and some of them can open and close due to a stimulus. Some of them just remain open all the time and allow for things to diffuse through. We also have carrier proteins, which can actually grab a hold of things, change its shape, and dump them into or out of the cell. So down here we have an example of a carrier protein. This one happens to be open all the time, so things are moving from an area of high concentration to an area of low concentration. So it is diffusion, but it's facilitated by this protein. Here we have a carrier protein down here where you can see it is diffusion, moving from area of high concentration to an area of low concentration, but it has to deform and change its shape to bring things in. There are other kinds of channel proteins that open and close due to a stimulus, and we'll talk about these when we do the nervous system and the muscular system. So some of these proteins can be uniports, where they only work in one direction. Some of them are symports, where they can work in where they can basically carry two things at one time in one direction. And sometimes there are the proteins that serve as antiports, where you can get substances moving in opposite directions. Okay, so we call these the symport and antiport, we call it co-transport, because the movement of one ion or molecule causes the movement of the other. And in symport, it's in the same direction. In antiport, we have basically the movement, the co-movement of things. One comes in, one goes out, but they move together through this protein. And uniport is just moving one substance in one direction. Now, active transport is another kind of transport mechanism. This one we're going to call primary active transport. There is a secondary, but we'll talk about that when we get into the digestive system. So primary active transport describes when a cell uses its own energy to pump an ion or a molecule across the cell membrane against its gradient. So it literally moves things from an area of low concentration to an area of high concentration, and in order to do that, you have to use energy to do that. This energy is provided in the form of ATP, and this mechanism is done by proteins that are in the membranes of your cells. These proteins will help to maintain or change the membrane potential. That is the voltage of the interior of the cell, at least right underneath the cell membrane, compared to the extracellular fluid that's around the cell. So this is a very critical thing, being able to change this membrane potential. And you'll get to know that well, because we'll talk a lot about membrane potential and changing it and maintaining it. So the sodium-potassium pump is our primary active transport protein. And you can see the proteins right here. It's a very, very complex protein. So look, we can start anywhere in the cycle. Let's start here in the cycle. So in this cycle of doing primary active transport, you can see the protein opens up, and in the interior it picks up 3 sodium ions. So they sit in these little spaces right here, 1, 2, 3. And because ATP joins with this protein, it actually changes the shape of the protein. That energy, when it drops a phosphate group off, it actually changes the shape of the protein. And so we can see the protein change its shape, and it dumps the sodium out. So 3 sodium were taken from the inside, and 3 sodium were dropped to the outside. Now the protein, because of its shape, after it drops the sodium off, this phosphate group will actually detach, and potassium, only 2 potassium will join. So those 2 potassium, once that phosphate disassociates, it's dropped. The potassium's dropped to the inside of the cell. Okay, so what you have is a mechanism here where you drop 3 sodium out, and you bring 2 potassium in. And you can do the math. You're losing 3 positive charges when you're only bringing 2 positive charges in. So that's going to, in addition to other things, help to maintain a slightly negative charge on the inside of the cell. So because you're dumping more positive out, the outside of the cell has more positive charge. So this difference between the positive and negative is that membrane potential. Okay. And there's a video that you can watch, but you can look at that online. I'll leave that video online for you to take a look at. So there's a little bit more to this picture here, and I wanted to kind of sketch out these little leak channels that form inside of the cell membrane. So let's just draw some little leak channels here. So the little rectangles are going to represent potassium leak channels. These little hourglass-shaped proteins are going to represent sodium leak channels. So here's another potassium leak channel. And then the circle is going to represent the sodium-potassium pump. This little line right here is representing the phospholipid bilayer membrane that you're associated with. In that membrane are these leak channels, though. So this is the potassium leak channel. This is the sodium leak channel. This is a potassium leak channel, and this is the sodium-potassium pump. Okay, so when you look at the membrane potential, it actually can be read. So if this were time and this were millivolts and this were negative 70 millivolts and maybe positive 40 millivolts, the cell is going to maintain a charge right on the interior of it of being negative 70 millivolts. So it's got a negative charge on the inside of the cell. So you can think of as the inner cell, this little cell membrane here, right underneath the cell membrane is negative. The fluids around the cell are going to be slightly positive. Okay, and that difference between that's the membrane potential. Now I can change that membrane potential. I can take and change that membrane potential and do something like this. So I can change the charge of it, but most cells are at rest or at negative 70 millivolts. So these potassium leak channels work this way because there's more potassium inside the cell than there is outside the cell. And the sodium leak channels work this way. Now these things are always open. So you always have a little bit of sodium leaking out, but you have a lot more potassium leaking out because there's a lot more potassium leak channels. And remember, the sodium-potassium pump, you're dumping out 3 sodium ions for every 2 potassium that you bring in. So you're constantly losing losing positive charge, and we're gaining very little positive charge back in. So these leak channels are really important to know about. The sodium leak channels, the potassium leak channels, the sodium-potassium pump, that helps to maintain the charge of the cell at negative 70 millivolts. If you change that charge, you can really do some damage to the cell. You can have water flow in and blow the cell up. And if you can't change the charge in nerve cells and muscle cells, if you can't cause the inside to become positive, then you can't send messages. So I'm just introducing leak channels here to you in the sodium-potassium pump so that you can get a better understanding of how muscles and nerve cells work when we get a chance to talk about those. And this is just a picture showing you regular old diffusion. So this is movement of from an area of high concentration to an area of low concentration, not using energy. Facilitated diffusion moves these ions from an area of low concentration, from an area of high concentration to an area of low concentration, but it uses a protein. It's facilitated by a protein. So active transport uses a protein. It uses energy to transport molecules from an area of low concentration to an area of high concentration. And that's going to be considered to be active transport. So the transport of large molecules also has very specific mechanisms too. The name of bringing in large molecules is endocytosis. And endocytosis, there is what we already talked about, phagocytosis. This is considered to be cellular eating. Penicytosis is cellular drinking. So your cells do gulp little amounts of water, and that's called penicytosis. And then there is a cell-mediated or receptor-mediated endocytosis, and this occurs when a molecule binds to a very specific receptor on the membrane, and then that very particular molecule is brought into the cell. And this is what they look like. So here is phagocytosis or cellular eating. Here's penicytosis. Instead of making big vacuoles, you make little itty-bitty vesicles. So that's how we bring in some substances and water. And then receptor-mediated endocytosis is where you have very specific receptors on the surface that join with very specific materials. And you can see that they can invaginate and then bring those materials into the cell. A way that we get rid of materials is exocytosis. So if we have the endoplasmic reticulum making something, it sends it to the Golgi apparatus, and then the Golgi apparatus sends it to the membrane, and it fuses with the membrane, thus dumping the products to the outside of the cell. And that's called exocytosis. Transcytosis is the third mechanism, and this is the transporting the substances in through and then in to the cell, through the cell, and then to the outs and to the other side of the cell. So sometimes we want to move product A from here to here, and the way we do that is through transcytosis. Our last little concept that we want to talk about, cell division, and this will be our last concept that we'll talk about with cells. So cell division is really important inside of cells because it's how we make new cells, how we replace cells, and how we reproduce cells. So the cell cycle is consisting of various stages. So after a cell goes through cell division, it's in what we call gap 1. In gap 1, this is the first little stage of cell's little life. And during that, if the cell is going to be a cell that's going to undergo cell division, it's going to basically take and duplicate all of its contents. It's going to grow in size. I like to think of gap 1 as the growth 1 stage. Now, not all cells in your body go through a stage called gap 1. They actually go into what we call gap 0. And this is a cell cycle stage where their cell cycle is arrested. They're not going to be actively dividing cells. And muscle cells and nerve cells, nerve cells are going to be cells that are in gap 0. And they just sit, they do work, but they don't divide. And most of the cells in your body do divide. So if they do divide, they go through gap 1. They then go through the S stage. This is where all of the genetic material is duplicated. So your 46 chromosomes are duplicated. So there's a double set of chromosomes. It then goes through a gap 2 phase where more growth occurs, and you have checks for errors. Your cells will make sure that the chromosomes are in the right number, and they make any kind of repairs they need to before they go through cell division. Cell division of animal cells, normal cell division in animal cells is called mitosis. And in mitosis, you go through different stages that we'll talk about in just a second, and then you form 2 daughter cells. These daughter cells may go into gap 1 or they may go into gap 0, depending upon if they're dividing cells or if they're non-dividing cells. The cell cycle is pretty important to know about too because of cancer. Cancer is where you have uncontrolled cell division, so the checks and balances through the course of the cell cycle is broken, and you get uncontrollable growth of cells. So the cell cycle includes various stages. So interphase is what we just talked about, where you had gap 1, S, and gap 2. That's all stages of interphase. If a cell is in gap 0, it's just in continuous interphase. So in prophase, this is the first stage of the cell cycle. You can see that the genetic material actually condenses into a form where we can actually begin to see it. And the nucleus of the cell the nuclear membrane begins to actually fragment and break apart. In metaphase of the cell cycle of mitosis, the chromosomes line up in the middle. In anaphase of the cell cycle, the chromosomes begin to separate. So half the chromosomes go this way, half the chromosomes go this way, so that each cell gets a full complement of chromosomes. In telophase, you have the separation of the genetic material, and in late telophase, the nuclear membrane will begin to reform. Okay, and then the cells will either go into gap 0 or gap 1, depending upon if they're dividing or not. So those are just the stages of cell division. We'll talk about this some in lab. Okay, well that completes the video lecture 2 and completes the cell structure lecture. So make sure you're the excellent student that you know that you want to be. Hard work, good work ethic, consistent work, working on study guides, working on root words, and doing your reading. All of those things will make you an excellent student, and hopefully to get you that grade that you want. So until next time, I wish you a good day or night whenever you're watching this video. I'll see you next time.